So this video is going to be about photosynthesis, and I'm going to use the same models that we use for cellular respiration. Before we use these models to be a mitochondria, but I'm going to make a couple modifications and we're going to magically turn this into a chloroplast. So the first thing I'm going to do is I'm going to add this here, and then I'm going to add this over here. What these are photosystems. So this is photosystem 2, and this over here is photosystem 1. And what I'm going to do is I'm going to set up a few things. Photosystem 2, I'm going to put in some electrons. So I'm going to put some beams on each of these circles, each of which represents a pigment molecule. And I'm going to leave the electrons out of photosystem 1 for now, and you'll see why in a moment. But I am going to take an NADP plus and an H plus and put them in these active sites on this particular protein that happens to be right next to photosystem 1. Ideally, the protein would like to stick these two together, but it can't because that process is endergonic and it doesn't have the energy to do it just yet. Note that this membrane, because we are now considering this a chloroplast, we would call this the thylakoid membrane. This area out here would be the stroma, and we'd have to ignore this little bit of membrane here. That wouldn't be there. And then this space in here would be inside the thylakoid. Note that we have protons kind of floating around. There's a high concentration in the stroma, low concentration inside. And then we also have ATP synthase, just like in cellular respiration. It's got a binding site for ADP. It's got a binding site for phosphate, but it can't stick those two together yet because that process is endergonic and it doesn't have the energy to do that yet. So what we are going to do is we're going to start over here with photosystem 2. And what happens in photosystem 2 is it's going to absorb a very specific wavelength of light. Now, there's different pigments in the photosystem, and different pigments can absorb different wavelengths of light. So let's just assume that this particular pigment is able to absorb the specific wavelength of light that hits it. That's going to excite this electron to a higher energy level. So it's going to jump up to a higher energy level. I'll kind of raise it up to represent that. And then it's going to fall back down. And the energy of it falling back down is going to provide this electron with the energy it needs to jump up. That electron will fall back down, which provides the energy this one needs to rise up. I'm going to put this in this little box called the excited state. And what's special about this one is that this is the reaction center chlorophyll. So when that electron gets excited, it gets snatched in that excited state by this particular protein. So this electron is now a high energy electron. Now, before we pass that on, we need to replace the one we lost. The molecule that does that is water. So I have H2O, and water is going to donate an electron to replace the one we lost and break apart. And in the process, we have oxygen, which is just released as a waste product from the plant cell. This electron that was snatched in the high energy state is now going to be passed from protein to protein to protein, just like in cellular respiration. Just like in cellular respiration, the energy of that electron moving from protein to protein to protein is used to pump hydrogen ions against the gradient. And just like in cellular respiration, these hydrogen ions, since we have a high concentration in the stroma, they're going to pass through ATP synthase down their gradient. The exergonic movement of H plus drives the endergonic production of ATP. So ATP production is very similar to what happens in cellular respiration. But what happens to that electron? Well, in cellular respiration, oxygen is the final electron acceptor. Here, this electron is passed to photosystem 1. Photosystem 1 then absorbs a specific wavelength of light that excites the electron to this higher energy state, where it is then passed to NADP plus and used to make NADPH. So what we've accomplished now is we've used the energy from light to produce ATP and NADPH. We're now going to take these two molecules and use them to build a sugar. So we'll walk to this next table over here. And essentially, the Calvin cycle uses carbon dioxide to build a sugar. It's a much more complex process I'm going to represent here. I'm essentially just going to highlight the main thing that's happening. And the main thing that happens in the Calvin cycle is carbon dioxide molecules are linked together. If we're going to link these things together, though, we need energy and we need electrons, but that's no problem because we have ATP and we have NADPH from the light-dependent reactions. So these two compounds are going to be used to link carbon dioxide molecules together into this 3-carbon unit called glyceraldehyde 3-phosphate. And then that can happen again. We're going to link some more CO2s using more ATP and NADPH into glyceraldehyde 3-phosphate. And eventually, this is going to be used to form a 6-carbon compound called glucose. So now we have used the energy of light to make ATP and NADPH, the energy of ATP and NADPH to build a sugar.